Hi, I'm Eric Leroux, Chief Science Officer at Marama Labs, and in this short video I will demonstrate how you can use scatter-free absorption spectroscopy and the CloudSpec to quantify antibodies and RNA in lipid nanoparticles. So here we're trying to quantify how much RNA and antibody we have in context of targeted LNP development. The existing methods like ribogreen for RNA or CMS, ELISA, PCA assay are not ideal. They have problems, mostly time-consuming, complicated, expensive, and perhaps more important, they lack accuracy. A lot of those issues are overcome by using SFA and the CloudSpec. All right, and the workflow is fairly simple. You would measure your free antibody, then you would measure your unconjugated LNPs, then your conjugated LNPs, and then you can use the CloudSpec software to compute the concentrations of your RNA and antibody, as I will demonstrate right now. This is the problem that we're facing. We have a spectrum of the conjugated antibody, which is the sum of the contributions from the unconjugated antibody reference. So this is a deconvolution problem where those weights here are related to the respective concentrations. So when you do these measurements, you always need to be within the recommended range for absorption. And if your absorption or scattering is too high, you should just adjust your dilutions so that you are within this range. The two reference components you need here are, in this case, the unconjugated RNA LNPs, so essentially before conjugation. You should try to keep the dilution similar to the conjugation. It's not critical, but it's a bit more accurate. And usually you would know the RNA concentration because you would have quantified it with CloudSpec using the standard workflow for just RNA. In addition, you need a pure reference spectrum for your antibody, and ideally in the same buffer as your LNPs. And we recommend a target range of typically 100 to 200 micrograms per milliliter for the reference. This is much higher than what you've got in your conjugated LNPs, but this is to ensure that the convolution is as accurate as possible. You can measure this once for a given antibody and keep it in your CloudSpec database and reuse it anytime you need to do the convolutions. Same for the unconjugated LNPs. You can measure them once for a given type of RNA LNPs and then use them for all the conjugated RNA LNPs that you derive from the same type. The deconvolution algorithm in CloudSpec includes an offset, and it should be a very small number to account for baseline fluctuations. That gives more reproducible results. So how do we do this in practice in the CloudSpec software? Here I've got an example with 3 conjugated LNPs. You can see they are very similar, so it's 3 repeats of the same thing. We have the unconjugated LNPs here. It's a bit less in terms of RNA concentration, as you can see at 260 here. That's not a problem for the deconvolution. And here we have our pure antibody spectrum. So for this analysis, we need to jump to the Analyze tab and first enable the antibody deconvolution. Then you can select your pure antibody and specify here its concentration or extinction coefficient. Usually concentration is recommended. That's usually what you know. And here, you rather than select pure RNA, as you would do for RNA quantification, you will select your unconjugated RNA LNP. It should be very similar to the RNA spectrum. And if you know the concentration, you should know the concentration and specify it here. And note that we don't select any empty LNP or lipids because this unconjugated RNA LNP already contains the lipid component in exactly the same concentration as are in our conjugated ones. So the deconvolution will work best with those unconjugated without lipids. Now, in this box, you can select up to 10 loaded LNPs, as long as they are made with from the same unconjugated RNA LNPs and from the same antibody. We've got here 3 repeats of the same thing. And once you've got all this, you can just run the analysis. And just while it's running, you can note that you could do a deconvolution with pure RNA, empty LNP, and pure antibody. The software will do it. We don't recommend it in the context of antibody because the antibody signal is quite small and the accuracy of the results is much better if you have this unconjugated RNA LNP. So this is our results, and you might recognize the results table. It's very similar to the pure RNA quant. Each row here corresponds to one of our loaded LNPs. And in the column have the concentration of RNA, concentration of antibody, which are scaled by the dilution. So in this case, just one, but otherwise they would be scaled. You can click on the row to see a graphical representation of that deconvolution. The red dashed line here is the fit, and it should be on top of your blue line if the fit worked. The green line is your unconjugated RNA contribution, which looks like an RNA spectrum, unconjugated LNP. And here you have the antibody contribution, which you can see is very small here. And actually, in this case, we have this number is an important number, the RNA to antibody ratio. We don't want this to be too large, otherwise this spectrum here would be very, very small to the point where you can't see it here. So when this number is higher than 4, you will see warnings, and basically you lose a lot of accuracy in your results. If you click on additional results, you'll have similar things if you're used to this ODs. But here the additional thing is the offset in milli-OD. This should be a fairly small number, typically, and the R squared that tells you that the convolution worked well in this case. And if you have more measurements, you don't need to remeasure all this. If it's based on the same references, you can just add them here. We run the analysis to have the results here in your table. So going back to the presentation, in summary, we've demonstrated how the CloudSpec can use a simple workflow to compute RNA and antibody concentrations. The precision for this process is of the order of 3% for RNA, 10% for antibody. It's a bit less precise because the antibody contribution is quite small. And the range of applicability, we recommend the RNA to be between 4 and 20 micrograms per mil. And as I said earlier, for the antibody, what matters is the ratio between RNA and antibody, and we don't want this to be too large. So if it's larger than 4, antibody concentration is too low, and this means a lot less accuracy. If you have any questions, feel free to contact us on this emails, and you can also check our other videos about the CloudSpec. Thank you for listening.